I'm Dr. Fakir Ravani, and this is clinical microbiology, susceptibility testing, and systematic approach to infectious diseases. Here's an overview of the human immune system. We can think of the immune system as innate immunity and adaptive immunity. The components of innate immunity include physical and chemical barriers. So physical, you can think of the skin, which basically prevents pathogens from entering the body. And chemical barriers, you can think of components of the saliva, and perhaps most importantly, the gastric acids that basically kill pathogens. Other components of innate immunity include mucus, the complement system, and innate cells such as macrophages, dendritic cells, granulocytes, natural killer cells. In general, the innate immunity is non-specific or broadly specific molecular patterns. And the actual immune response is through cytokines and chemokines, which lead to inflammation, phagocytosis processes, the complement activation, as well as activation of effector cells. Now, innate immunity is extremely important because in healthy individuals with an intact immune system, many features constitutively are present. In other words, you don't really need prior infection or immunization in order to have innate immunity. And the response is immediate. So it takes typically minutes to hours for the innate immunity to kick in and fight infection. However, it only persists for a few days, so it doesn't really give you long-term protection or memory. That's where adaptive immunity comes in. So the components of adaptive immunity includes humoral immunity, such as B lymphocyte cells and antibodies, as well as cell-mediated immunity, such as T lymphocytes or T cells. So adaptive immunity is antigen specific to a given pathogen. So it's not very broad, it's very specific to specific infectious diseases. And the actual response is through activation, proliferation, and differentiation of antigen specific B and T lymphocytes. It involves secretion of antibodies that can fight pathogens, as well as effector activity of T lymphocytes, and of course generation of memory cells, which lead to long-term immunity. And these mechanisms get activated either through actual natural infection or through immunization. Now, the first time the body is exposed to a pathogen, the adaptive immunity will take several days to develop, and that's referred to as primary response. So primary is the first time the body sees that pathogen. However, once the immune system has adapted to that pathogen, the next time the body encounters that same pathogen, there will be a rapid response due to the memory that develops. So because of that, there is a long-term duration of adaptive immunity. Now, once the adaptive immunity clears an infection, there is a contraction after elimination of pathogen that typically happens in a few weeks after the infection is cleared. And what contraction means is essentially, through adaptive immunity, the body will develop a lot of antibodies. And then once the infection is cleared, these antibodies start to wear off. So the levels of antibodies after a few weeks go down to very low levels, and that's called contraction. However, because the memory cells remain, and there are memory B cells are the cells that make antibodies. So memory B cells will last months and years, and there are also memory T cells that will probably last for the entire lifetime. And so the next time the body is exposed to the pathogen, because of this long-term memory, the body is able to expand the antibody levels very quickly. Now let's take a look at the hematopoiesis process. We're not going to go into too much details on this. We just need to know what the end products are and what their roles are in the immune system. Of course, this all starts with the hematopoietic stem cells, and these stem cells essentially differentiate into different cells, and these are the cells that exist in the blood. So, in general, we have three different kinds of cells in blood. Of course, we have red blood cells. Red blood cells, another name for it is erythrocytes. We have platelets. Another name for platelets is thrombocytes. And then everything else that you see here are essentially white blood cells. And another word for white blood cells are leukocytes. So now, because white blood cells have an important role in fighting infections, we're going to go into more details of the different types of cells that constitute leukocytes. Essentially, anything on the right-hand side on this pathway are lymphocytes. So we have B lymphocytes, we have T lymphocytes, and we have natural killer cells. And then everything to the left essentially comes from myeloid progenitor. So these are basophils, eosinophils, neutrophils, mast cells, and monocytes. Now, when we measure white blood cells in the blood, we typically count how many cells are in a microliter of blood. So, the normal range is anywhere between 4,000 to 11,000 cells per microliter. Now, if somebody has low levels of white blood cell, that's referred to as leukopenia. And of course, the other end is if somebody has too much white blood cell, it's referred to as leukocytosis. And that typically happens when people have an infection. Not every infection will lead to increased white blood cell, but if it is a type of infection that requires white blood cells to fight it, the body will start making more white blood cells. So, a typical sign of an infection would be if somebody has elevated white blood cell or, more appropriately called leukocytosis. And that would be if somebody has a level 12,000 or higher. Now, here on the right-hand side, you can see the shorthand way of writing some of these lab values. So on the left hand side is white blood cell, on the right hand side is platelet, at the top is hemoglobin, and at the bottom is hematocrit. And this is a shorthand when typically clinicians, when they work up patients, this is how they write. So you don't have to write white blood cell, you just write numbers here instead of these labels. And typically, if somebody says white blood cell is 4, they really mean 4,000. They're not going to write 4,000 or 5,000. So they typically write, like 6.2 or, 13. So if somebody writes 13 here, they really mean 13,000. Now, if you know somebody has leukopenia and they write 50 here, they're really talking about 50 cells. They're not going to write 0.050. So you just have to use it in the context. The same with platelets. So, typically people ignore the 3 zeros at the end. Now, hemoglobin and hematocrits are more straightforward, so the numbers are not too large. They're not in the thousands. So when somebody writes here hemoglobin is 14, they actually mean 14. And hematocrit is in percentage, so that one is easy. Now let's take a look at the 6 basic types of leukocytes. So eosinophils constitute about 1 to 3% of all white blood cells. Basophils and mast cells, less than 1% of white blood cells. Neutrophils are extremely important. Neutrophils are the primary mechanism of fighting bacterial infection. So when it comes to bacteria, neutrophils have a primary role. And that's why you will see a large portion of white blood cells, typically 50 to 70% are neutrophils. Now, an important concept when it comes to neutrophil is the concept of segs and bands. So what we mean by segs or PMNs or polymorphonuclear leukocytes or just polys is that these are essentially mature neutrophils. And then the immature neutrophils are called bands, so band cells. And essentially, when the body makes new neutrophils, initially they are bands, and then it takes time for them to mature and become segs. So normally, most of the neutrophils circulating in blood are mature in the mature form, with the nucleus of the cell being divided or segmented. So that's where the name segs comes from. Now, during infection, because the body tries to make more neutrophils than usual, you will see an increase in the band or immature neutrophils, and that's typically referred to as bandemia or left shift. So left shift meaning the cells have shifted to the immature side. And so if you see more than 10% of the neutrophils in the band cell, that's typically called bandemia. So this percentage here is 3 to 5% of white blood cells are normally bands, and white blood cells, about half of them, 50 to 70% are white blood cells. So this 3 to 5%, you can think of it as typically 6 to 10% of neutrophils. So if you see more than 10% of neutrophils as bands, that's typically clinically significant. Now, other types of leukocytes include monocytes, and they actually progress to become macrophages, and those are 1 to 6% of white blood cells. And then we have lymphocytes and their derivative plasma cells, and a big portion of white blood cells are lymphocytes, and they have an important role in developing memory. But also they have a big role when it comes to viral infections, and of course they also important in other types of infections, including bacterial and fungal infections. And lastly, we have dendritic cells, less than 1% of white blood cells. Now here are some more details about the function of each leukocyte type. In general, eosinophils have a role in allergic reactions, including allergic reaction to antimicrobial agents. They also have a role in fighting parasitic infections. Basophils and mast cells are also active during allergic response, especially anything with histamine release, and they have a role in inflammation. As I mentioned, neutrophils have a big role in bacterial infection, but they're also important in fungal infections. Macrophages are not only important in bacterial infections, but they're also antigen presenting cells, so they activate lymphocytes. And of course, the three types of lymphocytes. B lymphocytes are the ones that result in plasma cells that make antibodies. B lymphocytes themselves are also antigen presenting cells, so they can work with T cells. T cells, there are different types. There are CD4, CD8 that are most commonly important types of T lymphocytes. They are not only important in developing long-lasting memory to bacterial infections, but they also have an important role in fighting viral infections. We have natural killer cells, and lastly, we have dendritic cells that are also antigen presenting cells. Please pause this video and review the function of each of these leukocyte types. Now that we have a good foundation, let's take a look at our first learning objective. Given the results of a specimen stain, list the most likely organisms present in the specimen. And the specimen would be from the site of infection. So this could be blood. If somebody has infection in the blood, referred to as bacteremia, or could be urine or sputum or CSF or other types of specimen. Let's take a look at different types of bacteria. So in general, bacteria are divided into different groups based on how the cell wall is stained. So if you look at bacteria under microscope, you're not going to see anything because they're transparent. So in order to see them under microscope, we have to use some techniques to stain them so we can see them. If we look at bacterial cells, you will see inside the cell, there is a genetic material, and then the cell is surrounded by a cytoplasmic membrane or the cell membrane. And then what you will see outside of the cell membrane is a cell wall, which is essentially made of peptidoglycans for most of these bacteria. And that's what we stain. So we stain the cell wall, and the cell wall is what gives the shape of the bacteria. So they will have different shapes. So, either rod shaped or spiral or other shapes that we will talk about shortly. And one stain that we can use is called the Gram stain. So Gram was, is the name of the person who came up with the technique. And some of these bacteria, when you use the Gram stain, they will turn blue, and we call that Gram positive. So something that's particular about Gram positive is that they have the cell membrane, and then outside of the cell membrane, they have a thick layer of peptidoglycan, and then this layer gets, this is the cell wall that gets stained blue. And then we have another group of bacteria that are called Gram negative. These essentially stain, pink using Gram staining. And something that's particular about the Gram negative is that, they still have the cell membrane followed by a thin layer of peptidoglycan. And the reason this is thin is that on top of the peptidoglycan, there's a second outer membrane. So there are two membranes. So that's a major difference between Gram positive and Gram negative, that, Gram negatives have this outer membrane. So the peptidoglycan is thinner. And then we have a third group that just doesn't get stained using the Gram technique. So then we will have to use a different type of staining. So the acid-fast staining is used. A good example is Mycobacteria. For example, if you think of tuberculosis. So these get stained with acid-fast. And then the fourth group is what we call atypical, because they do not have cell wall. So, they do not have peptidoglycan, because these three have peptidoglycan that can get, one way or another, they're going to get stained. But the atypicals, because they don't have a cell wall, they don't get stained. So, any of these techniques you use, they will not be stained. So these are atypical. So these are in general the four types of bacteria that are clinically important. Now let's take a look at some of these staining and microscopy techniques. So as I mentioned, the Gram stain is the most commonly used technique, and this technique can stain bacteria, but also some fungi species can also get stained. Certain bacteria, most importantly Mycobacteria, do not get stained using Gram stain, so acid-fast is what we use. And then there are specific techniques that are used when it comes to different types of fungal species. Now let's take a look at some specific examples of Gram-positive bacteria. Now in general, whether it's Gram-positive or Gram-negative, these bacteria can be either aerobic or anaerobic. So essentially aerobic means that they require oxygen for replication. Anaerobic means that they can replicate in the absence of oxygen. And these definitions get more nuanced from a microbiology standpoint, but for us, clinically, that will suffice to distinguish between aerobic and anaerobic. And then with Gram-positive specifically, we can look at morphology. So morphology is basically the shape of these and how they group together when you look at them under the microscope. So essentially, we can see cocci, and this is essentially in circular shape, and they can be in clusters. So cluster means that several of them are clustering together. And then you can have cocci, which is essentially circles in pairs or chains. So pairs will be a couple of them, and sometimes that's referred to as diplococci. And then, if you have a chain, they don't cluster. It's just, just like a chain. And then we have rod shape that are called bacillus, and the plural for it would be bacilli. And I should say cocci is a plural word. The singular form is coccus. So if you're talking about the single bacterium, it will be a coccus. And then so for Gram-positives that look like cocci in clusters, typically there's a test they do, then these tests catalase positive. And then on top of that, in the micro lab, they can do a coagulase test. And if you have coagulase positive, then you have a very important, clinically important organism, Staphylococcus aureus. Okay, and the reason we're talking about this is because it takes some time to identify what the organism is. And if somebody has an infection and we don't know what it is yet, but they look at it under the microscope and they say, well, it looks like cocci in clusters, and we also did a coagulase test and coagulase test was positive, as clinicians we can anticipate Staphylococcus aureus, so we can give appropriate treatment. So that's why we're covering this. We're not trying to become microbiologists, but as clinicians you can use this clinically to help patients. And if they say the coagulase is negative, you have several options, and together all of these are referred to as coagulase negative staph or coagulase negative staphylococcus. And there are several species that are coag negative, and I have listed 4 that are more common. And Staphylococcus lugdunensis is one that can cause severe infection, so that's why I have bolded it. But so these are typically present on the skin. The same with Staphylococcus aureus. All staphylococci are typically present on skin, and occasionally they find their way into the body and cause infection. And then for the catalase negative group, which they do not look like a cluster, so they will be in chain or maybe pairs. Then we can do the hemolysis test. So what a hemolysis test would be is that essentially an agar is essentially a plate. So what we do is that we use sheep blood, and we basically let these organisms grow and see what they do to the blood cells. So the beta hemolytic group, which you can see in this picture, essentially they lyse blood cells, so they will break it down and it will basically clear the blood, and that's called beta hemolytic strep. And the alpha essentially reduce hemoglobin and they cause this greenish color. And then there is a third group that don't really do anything to the blood, and these are referred to as non-hemolytic because they didn't break it down. So essentially that's how we group these. So beta hemolytic strep, alpha hemolytic strep, and non-hemolytic. So in the beta hemolytic group, we have 2 important Streptococcus species. So Strep pyogenes, which is sometimes referred to as group A strep. And then we have Streptococcus agalactiae, which is referred to as group B strep. So these are important, and you should know these interchangeably, because sometimes people may refer to them as group B strep. So they might say, okay, for this patient we are trying to cover group B strep, so you should know that they are referring to Streptococcus agalactiae. And if they say we want to cover group A strep, you should know they're talking about Streptococcus pyogenes. And then for the alpha hemolytic group, the most important is Streptococcus pneumoniae. This is very important because it causes respiratory infections, and we'll cover this in detail throughout the course. And then there's also the viridans group Streptococci. When it says group, there are multiple species, and there's really no need to know specific species. They're collectively referred to as viridans group Streptococci. And then the non-hemolytic, we have 2 Enterococcus. In the old days, these were also called Streptococcus, but nomenclature has changed over time. So nowadays, these are called Enterococcus, but they're also in the same group as the rest of the strep. Enterococcus faecalis and Enterococcus faecium, these are 2 organisms that are kind of the frenemy, so they sometimes they are good because they can be as part of the flora in the body and do some good, and sometimes they can be bad and they can cause an infection. So that's why they're tricky. So you have, and we'll cover this throughout the course. So sometimes For example, they might be in the urine and prevent urinary tract infection. Other times they might be the cause of urinary infection. So it becomes tricky when we identify this in a specimen. So a lot of caution should be used. And then lastly, these are just the gram-positive bacilli, so Listeria, Bacillus anthracis, and Corynebacterium diphtheria. Now let's take a look at gram-negative bacteria. Again, we're focusing on aerobic for now. So you will see that majority of gram-negative are actually rod-shaped. So these are bacilli. And what we do with gram-negatives is that we do a lactose fermenting test. So if they can ferment lactose, we call them, actually the important thing is to identify what's not fermenting lactose, and they're typically referred to as non-fermenters, okay? But if they do ferment lactose, then, they do oxidase tests, and, for this course, you don't have to worry about the oxidase test, but know that when it's lactose fermenting, for the most part, we're talking about Enterobacterialis, and Enterobacterialis is a family of several organisms, and Enterobacterialis used to be called Enterobacteriaceae. And the important organisms in Enterobacterialis family is Escherichia coli or E. coli, Klebsiella species. So when it says SPP, that means there are multiple. So for example, there is Klebsiella pneumoniae and there is Klebsiella oxytoca. And there is Enterobacter cloaceae. There are several Citrobacter species. There are Serratia species, Providentia species, and Protease species. So these are all under Enterobacterialis. And then we have, other lactose fermenters, so Aeromonas, Pasturella, and Vibrio species. And then when it comes to non-lactose fermenters, and this is bolder because these are very important organisms because they typically are very resistant to a lot of antibiotics. So the most important would be Pseudomonas aeruginosa. So this causes a lot of severe infections and becomes difficult to treat. And also Acinetobacter baumannii, which is less severe than Pseudomonas but can be more resistant to a lot of organisms. And then, Stenotrophomonas and Burkholderia also can be very, very resistant. And this typically, Stenotrophomonas and Burkholderia typically more common in immunocompromised patients. And then for the cocci in the gram-negative group, we have Neisseria meningitidis, Neisseria gonorrhoeae, and then we have Haemophilus influenzae and Moraxella catarrhalis. These two are actually kind of between cocci and bacilli, so they're referred to as coccobacilli. So the shape under microscope is between a circle and a rod. And then spirochetes, which are essentially look like a spring. So we have a couple of them, Troponema pallidum, as well as Borrelia burgdorferi. Now let's take a look at some anaerobes. So anaerobes, gram-positive is Peptostreptococcus, and then we have a series of Clostridium and Clostridioides difficile. So Clostridioides difficile also used to be called Clostridium difficile, and nomenclature changed a few years ago. And we will cover these later in the course. And the gram-negative anaerobes, essentially the most important you need to know is Bacteroides, because any intra-abdominal infection, Bacteroides have an important role. Atypical bacteria include chlamydia species, including chlamydia pneumoniae. That's not a sexually transmitted infection. It can cause, pneumonia. And then we have the sexually transmitted chlamydia. Also, we have Mycoplasma and Legionella. And then with the acid fast, we have nocardia species, we have Mycobacteria, as well as Actinomyces. Looking at the results of a specimen stain, we listed the most likely organisms present in the specimen. Now let's describe the basic process of microbiology testing for identification and susceptibility. Let's take a look at the process of microbiology testing when a patient who's suspected to have an infection presents. So when the patient presents with symptoms and signs of an infection, we'll call this time 0. And typically, it will take anywhere between 1 to 3 hours for clinicians to examine the patient and, if necessary, collect specimens. And these specimens will be from the site of infection. For example, it could be blood or urine or any other specimen. And then the specimen will be sent to the lab, to the microbiology lab for testing. And what happens at the microbiology lab is that you typically have to incubate these specimens in order for bacteria to grow before we can detect anything. And this can be done using automated systems, or it can be done on an actual plate. So these plates are called agar plates. And what they are is that they essentially have nutrients in them for bacteria to grow, and you just let them sit there and incubate under the right temperature for bacteria to grow. And then after a while, typically anywhere between a few hours to a few days, depending on the organism, we may start to find colonies of the organism. Now, of course, it's possible that nothing may grow. So, sometimes it will be difficult to identify. But if there is a specific organism that will be identified, it will be growing typically between 12 hours to 5 days. And depending on what type of infection we are suspecting in the patient, if we think that it might be an organism that's a very slow grower, it will be wise to keep the specimen for beyond 5 days in order to give it enough time to grow. Now, if something does grow, that's when, typically they start to stain it and then look at it under the microscope. So while the process of staining, is not time consuming, now if you look at the entire process, it typically takes somewhere between, half a day to a day, if not more, before they can actually look at it, stain it, and identify, if it's bacterial infection, whether it's gram-positive, gram-negative, or atypical, and then look under microscope to look at morphology. And then there are a series of tests that will be done in the micro lab in order to identify. So this is where they do those, the lactase test and oxidase test and hemolysis test. And so there will be numerous tests that will be done if applicable, in order to actually identify what the exact organism is. And that typically takes anywhere between 1 to 2 days after staining. So now, this is typically day 2 since that patient presented at the minimum. And once an organism is identified, so for example, if they say this is E. coli, then the next step would be to identify antimicrobial susceptibility. So then, it typically takes another 24 to 48 hours to do various testing, and we will go over some of these susceptibility testing. But the key point here is that it will take, another day or two after the organism is identified in order to find susceptibility results. And what that means is that, they will tell you like which antibiotics will be active against this organism and which ones will not be active, so that we can streamline treatment. Now, as technology advances, we have a rapid diagnostic test that can expedite this process. So, when we actually grow an organism, we can directly put the some of these colonies into rapid diagnostic tests, and these rapid diagnostic tests will be able to very quickly, in a matter of anywhere between under an hour to just 2 or 3 hours, tell us exactly what the organism is and what the susceptibility results are. Now the problem is these diagnostic tests are limited to only a select few organisms, so they're not comprehensive of every possibility. So it's not a replacement for the traditional process. So even though we used a rapid diagnostic test, we still need to do the traditional identification and susceptibility testing, because in case the patient has an organism that is not included in this rapid diagnostic test, we still need to figure it out, and you don't want to slow down this process while the rapid diagnostic test is processing. But certainly these rapid diagnostic tests are advancing very quickly year after year. Now, what happens in parallel to this process is what we do to treat the patient, because we're not going to wait for this process to complete and then start treating the patient, because the patient could die by day 3 when we find out exactly what antibiotic will work. So what we do is initially we do empiric antimicrobial therapy. So what empiric means is that we don't really know what's causing the infection, but we will do our best guess to, based on the signs and symptoms, we can anticipate what organisms are possible, and we can use antibiotics broad enough to cover the possibilities. So now we can kind of look at the timeline of treatment. So we typically, we want to avoid treatment as soon as the patient arrives, because if you give the antibiotics immediately, you might not be able to identify anything because you kill all the organisms. So you want to get your samples first if possible, and then start treatment after you get the samples. And then, once we get rapid diagnostic results, that's your first opportunity to optimize treatment. So based on what you find, you may be able to change the antibiotics to have something that's better for the patient. The same with, when you have gram stain results, you can change the antibiotics. And ultimately, when antimicrobial susceptibility results are available, at that point we will switch from empiric therapy to pathogen directed therapy, meaning that we know exactly what is the best antibiotic for the patient that will be active against the organism and will be the narrowest that would get to the site of infection and will result in the optimal outcome for the patient. When it comes to phenotypic susceptibility testing, the concept of MIC is extremely important. So MIC stands for minimum inhibitory concentration, and it is the lowest concentration of an antibiotic that inhibits visible growth of a microorganism. And that's why, we say a phenotype is what actually appears. So, it will be visible to the actual human eyes. And that's to distinguish it from genotypic susceptibility testing. So this is, regardless of what resistant genes may be present in an organism, the minimum inhibitory concentration just, shows you what's the minimum concentration to inhibit growth of organism. And there are different ways to measure MIC. So the gold standard is the broth microdilution, and by gold standard I mean that this would be the reference method, and any other method would be something to as a surrogate for the broth microdilution. And here's how broth microdilution works. So you can kind of imagine a series of test tubes in the microbiology lab, and then in each test tube we will have a broth. So what broth is essentially nutrients for bacteria to growth. Now these are sterile, so there are no bacteria in them, and what we do is that we put antibiotic of choice that we are trying to test in these test tubes, and we increase the concentration of antibiotic in each test tube, so kind of double the dilution. So essentially one of them will not have any antibiotics, so the concentration would be 0, and then we kind of double the concentration in each test tube, and then we inoculate these test tubes with the organism that we are trying to test, and we let it grow. And then after the incubation period is over, we can see that it actually, the growth becomes visible. So you can see that it has become cloudy, or the color may have changed. And then you can see at what concentration nothing grew. So for example, in this example, you can see that at the concentration of 64 and above, nothing was growing. So the minimum concentration was needed to kill the organism in this example is 64, because a concentration of 32, the organism was growing in the test tube, but at 64 or higher, nothing was growing in the test tube. And that's how they measure the minimum inhibitory concentration for a specific antibiotic. And MICs, so every antibiotic has a different MIC for each organism that it covers. So if you have antibiotic A, it will have MIC for E. coli, let's say. It will also have a different MIC for Klebsiella, and so forth, if the antibiotic A is active against these organisms. And then the same if when you go to a different antibiotic, you have to measure the MIC for that different antibiotic for each organism. Now, another method to do is disk diffusion, also referred to as Kirby-Bauer method. And the way this works is that essentially, instead of test tubes, which you can imagine that it will be very labor intensive, so broth microdilution takes a lot of space and requires a lot of labor, so it may not be feasible. And that's why we say it's a reference method. So a much more feasible method would be disk diffusion, and here's how it works. So instead of test tubes, you have an agar plate. So again, this plate is sterile. What's on the plate is nutrients for bacteria to grow. And what we do is that you inoculate the agar plate with the organism that we're trying to test, and then immediately after we inoculate the plate, we place several discs. So these discs are essentially different antibiotics. So each disc has a certain concentration of the antibiotic. And when you place these discs in the region near each disc, this antibiotic starts to diffuse. And the further you go from the center of the disc, the concentration drops. Okay, and then at some point when you go too far from that disc, that specific antibiotic may not be present. So each disc has a different antibiotics. So you are simultaneously testing multiple antibiotics for this particular organism. And then if you have multiple organisms, then you will have one plate for each organism. So you can have several plates set up. And then you let it grow. So after the incubation period, you kind of can see that there are there is essentially a lawn of the organism that grows. And if the organism is susceptible to that antibiotic, there will be a zone of inhibition. In other words, because antibiotics have diffused in this region, the organism will not be growing. And if it's resistant to it, you can see that the organism will grow through the disc. Essentially means that antibiotic was not active. And then we can kind of correlate this to some, whether it's, is it really susceptible, or is there some sort of intermediate resistance going on? So we can measure the zone of inhibition. So you can see this is a circle. They can actually measure what's the diameter of this zone, because as you can see, some of them have a smaller diameter, others have larger diameter. So the larger the diameter means the more active the antibiotic is. And we'll talk about how to interpret this zone of inhibition shortly. And of course, if it goes through the disc, that means that the organism is completely resistant to that antibiotic. So that's disk diffusion. And then another method, disk diffusion only tells you if it's susceptible, intermediate, or resistant. It doesn't really give you MIC. Okay, so broth microdilution will give you MIC, and then based on the MIC, we can decide if an organism is susceptible, intermediate, or resistant to the given antibiotic. This diffusion only tells you if it's susceptible, intermediate, or resistant without the MIC. So another way to do it in order to get the MIC is the e-test. So the way e-test works is similar to disk diffusion. The difference is that instead of placing disks and then measuring the zone of inhibition, you put a test strip. So there is a strip that has a given antibiotic at different concentrations. So you can see that, at the top, it has a very high concentration of antibiotics, and the more you go down the strip, the concentration drops. And then, again, you inoculate the plate, you place the strip, and you let it incubate. And then after incubation, you can kind of see a zone of inhibition, and then you can see at what concentration the antibiotic stopped inhibiting growth. So that would be your MIC. So in this example, we can see that it stopped growing at concentration of 0.126 or higher. So the MIC would be that number, 0.126. So that's something that's also done in practice. And another way that's more commonly used is the automated machine. So there are certain machines that are automated, essentially. So you can see the one example in this picture where you essentially load this with your sample. You can test a lot of samples simultaneously. So there are these cartridges that you put in, and you just run the machine, and it may take some time for the machine to run. And then, in a matter of an hour or so, and then it will actually give you MICs. So then you can, get your MIC and interpret it as susceptible, intermediate, or resistant. And this is, these automated systems are most commonly used for majority of the samples. Now, the issue is that because these machines are difficult to make, they typically come preloaded with certain antibiotics. So if a new antibiotic comes to the market, it may not be available in this machine, and that's where e-test comes in. So anytime a new antibiotic comes to the market, the manufacturers will make e-tests available so that micro labs can test for susceptibility so they can use the new antibiotic. Now, when you get your MIC results, or if you get results from this diffusion, what they really tell you is, those MICs are essentially in a test tube or on a plate, which we call in vitro, meaning it's not inside a living host. Although we know there is a minimum inhibitory concentration, that doesn't necessarily mean that we would be able to achieve that concentration inside the patient's body. So, it has to do with, the dosing of antibiotics that are safe. Would we be able to achieve that concentration in the patient's body? So in order to interpret that MICs and see if it's feasible to use that antibiotic, there are criteria. So just knowing the MIC doesn't really tell you whether you can use it or not until you interpret it. And the way we interpret it is that we use CLSI is the organization that gives us the interpretive criteria or breakpoints for MICs. And and I will show you how to navigate this document. But essentially, they will tell you for a given organism, for each antibiotic, they will tell you at what MIC cutoffs it will be considered susceptible, intermediate, resistant, and for some of these there will be a susceptible dose dependent, meaning that it would be susceptible as long as you give a certain amount of antibiotic. So if you click the link, you will go to CLSI's website where they will host the M100 document. The M100 document is the one that has interpretive criteria for all antibiotics. So it doesn't have antifungals or antivirals. It's specifically gram positive and gram negative organisms. And then this is available for free, so you can just click to enter as a guest, and then you just make sure you click on M100 document, and this gets updated annually. So for example, next year you will see the 2023 edition. So keep that in mind. And then once you go here, you can use the table of contents, and there is a table for each organism. So if you scroll down, you will see that starting with table 2A, so this one 2A is for Enterobacterialis, and then the next table is for Pseudomonas, the next one is for Acinetobacter, next one is so on and so forth. So if you are looking for breakpoints for specific organism, we're going to use Enterobacterialis for this example. So if you click Enterobacterialis, it will take you to the page for Enterobacterialis. And this document is intended for microbiology lab staff. So a lot of this you can ignore. The part that you need when you look at the breakpoints is the table. So you will see that it has columns for susceptible, dose-dependent, intermediate, and resistant. And there are two sets of them. So one of them is based on MIC. So if you are using an automated machine that gives you MIC, or if you're using e-test, this is the part you will use. And if you're using this diffusion where the diameter of zone of inhibition is important, they will tell you, how big does the zone need to be. So in this example, let's say if you have E. coli as the organism and you're testing against ampicillin in this diffusion, they're telling you that if you measure the diameter and if at least 17 millimeter, it will be susceptible. Now, if it's between 14 and 16 millimeters, it will be intermediate. And if the zone is smaller than 13 or less, it will be resistant, including, if the zone is 0, which means that it just grew through the disc. Now, you guys don't need to memorize any of these numbers. I'm just trying to show you how to navigate and use this document. Now, with MIC, so again, for susceptible, it will say MIC of 8 or less is susceptible. So in other words, if you test it and MIC comes back and it says MIC is 4, you look at here and say, oh, good, it's 8 or less. So, for that patient, it would be susceptible. But if you had, another test result come back and said the MIC is 16, you look here and say, oh, it's 16. So it's not susceptible and it's not resistant and it's intermediate. And what intermediate means is that if you're using the usual approved doses of ampicillin, it will likely not work. But if you were to use higher doses, it will probably work. And if it's resistant, doesn't matter. If you use the maximum dose that's safe in patients, it's just not going to work. So you do not want to use ampicillin if it's resistant. And so if the MIC comes back and it's 32, it's resistant. If it's 64, it's resistant. Now, with intermediate, also in general, if something is intermediate, we try to avoid it if we have other options. So occasionally, you may not have other options available. So in situations like that, we will have no choice but to use it if it's intermediate. But if it's resistant, under no circumstances will we be using that antibiotic. In this case, susceptible dose dependent doesn't exist. So susceptible dose dependent only exists for certain antibodies. So for example, a good example is cefepime. So if you go down to cefepime, which is a cephalosporin, you will see that under the column for susceptible dose dependent, they have listed 4 to 8. So this means that if you have, for example, Klebsiella pneumoniae, which is a member of Enterobacterialis, and you get an MIC for cefepime back and it's 2, you're going to say, okay, good, 2 is susceptible. Now, if it comes back and it's 8, then you're going to look here and say, oh, it's 8. It's not susceptible, but it's susceptible dose dependent. And then you can look at the notes here. So what does that mean, dose dependent? And then they will tell you that essentially, if it's susceptible, it's based on if you're using a dose of 1 gram every 12 hours. But if it's 4 or 8, you need a higher dose of higher than 1 gram every 12 hours. And they guide you to Appendix E, where they have a specific recommendation. And essentially you will see in that part that, if it's MIC of 4, you need to have 1 gram Q8 or 2 gram Q12. And if it's MIC of 8, you need to use the maximum dose, which is 2 gram Q8. Maximum, as far as FDA approved maximum dose. And of course, if it's 16, it will be resistant. And because there is a susceptible dose dependent, then there is no intermediate. So they have eliminated the intermediate for this antibiotic. So that's how you navigate the CLSI document. Here's another example. So let's say we have identified E. coli, and then we did broth microdilution to see the MIC. So, looking at this, you can see that this test tube, this is cloudy, and these 3 are clear. So which means that organism E. coli was growing in these 2, but not here, here, or here. So the minimum concentration needed to kill E. coli in this example was 8. So MIC is 8. Now, just knowing the MIC alone doesn't tell you anything of what you can do. And I should say this is ciprofloxacin in these test tubes. So just knowing MIC is 8 doesn't mean if we can use ciprofloxacin or not. So what we need to do is to interpret this MIC. So CLSI document, I put a screenshot of the table here. You can see that for MIC, they say to be susceptible, it has to be less than or equal to 0.25, and then anything greater than or equal to 1 is resistant. In this case is 8, so it's definitely resistant. So this E. coli in this sample is resistant to ciprofloxacin. Even though, if you give high enough of ciprofloxacin, it's going to kill it, right? But it's considered resistant because we cannot given the safe doses of ciprofloxacin, we will not be able to achieve these concentrations in the human body. So we will consider resistant and not use ciprofloxacin for a patient who has this isolate. Okay, that was phenotypic testing. Now, when it comes to genotypes and testing for genes, we have rapid diagnostic tests, sometimes called molecular tests. And, some of the common ones is the PCR test, so multiplex PCR. There are different tests out there. So one example is the BioFire film array BCID2. So this test that was developed in recent time can detect any of these organisms listed here. These are the ones that are commonly encountered in clinical practice and are problematic. So it's good to identify these rapidly. Now, if it doesn't detect any of these, doesn't mean that the patient doesn't have an infection. It just means that it could be something that's not on the list. And of course, occasionally there could be a false negative, right? So that, it could be that somebody has one of these and the test just couldn't detect it. And that's extremely rare because these tests have an extremely high sensitivity. And then the important thing about these PCR tests is that it can also detect these genes. So these are genes that we will cover in a different topic on another lecture. But if these are detected, that means that the organism can be resistant to certain antibiotics, and we'll cover what those antibiotics are later. And also, this is really fast. So this will give you results within an hour once you run the PCR. And of course, you have to wait, probably around half a day to a day for the organism to grow first. But once you have an isolate, then you put it in the PCR, and then within an hour, it will give you the results. Another test is the Verigene test that's commonly used. There are 2 different. They have one specifically for gram-positive and one for gram-negative. So these are, again, the organisms that it's capable of detecting and the resistant genes that go with them. And this one takes a bit longer, but still pretty reasonable. It will tell you the results in about 2 hours, 2 to 2.5 hours. And then beyond PCR, we have other technology. So MALDI-TOF is something that can essentially detect any possible organism. So this is a really powerful machine that can detect any possibility. and it's pretty fast, so within 30 minutes. Now the downside is that it cannot reliably detect resistance, and there's a question mark here because it's a work it's under development, so there's a lot of research going on for MALDI-TOF to reliably report susceptibility as well. But currently pretty much it's only used for, in most places, for detecting organism but not resistance. And then we have pinnafish. There are different types, and these are also pretty fast within 20 minutes, and again, not reliable for detecting resistance, but they can detect some organisms that are listed under them. And the most recent technology is the NMR. So there's a T2 bacteria. So this one is really interesting because it goes directly from blood. So if you think of the whole process of microbiology testing, so when a patient comes in, we get a sample, and then you got to incubate it, and then once you have and isolate, then you can do rapid diagnostic testing, right? With the T2, when it says direct from blood, that means if you got blood samples from the patient, it goes directly from here to rapid diagnostic test. So you save, half a day or so. So that's the big thing with the NMR technology. So it goes directly from whole blood. Now, unfortunately, currently it cannot detect resistance, but it can detect these organisms pretty quickly. We interpreted the results of a specimen stain to list the most likely organisms present in the specimen. We described the basic process of microbiology testing for identification and susceptibility. Now, let's identify the normal human flora and their role in infectious diseases. Now, normal flora are absolutely essential for health. There are non-sterile sites in the human body where these flora reside. However, there are sterile sites that are not supposed to have any organisms in healthy hosts. So in general, all internal organs are considered sterile. So they should not have any flora. For example, in blood, we don't anticipate to have any organisms. So if you do find an organism in this sterile site, they're considered in infection for the most part. So especially blood, if you find anything in the blood, it's considered infectious. The same with the central nervous system, cerebral spinal fluid, in the liver, spleen, kidneys, lower bronchi and alveoli, and the bladder for the most part. So the bladder is a bit tricky because especially in young men, the bladder is supposed to be sterile. But as part of aging, for most people, as we get older, we start to have colonization of bladder. That may, as people get older, bladder is no longer sterile. But a young man, it will definitely should be sterile. In women, it's common for women, given the anatomical differences between men and women, that the bladder will be colonized, and women, as they get older, also the percentage of colonization goes up. And anybody who has a urinary catheter almost certainly has colonization of bladder. So the bladder is kind of tricky on this list, but the rest of this list is pretty much, if you find an organism, it's likely due to infection. But let's focus on the normal flora in these other sites. So the skin in general has gram-positive aerobes, such as any Staphylococci species, including Staph aureus. So if you find Staph aureus on the skin, it's not necessarily infection in a healthy host. Occasionally, this may cause skin infection. But in the absence of any signs and symptoms of an infection, it's okay to have these organisms on the skin. That's part of the flora. There are obviously oral flora in the mouth. So, Fusobacterium, Peptostreptococcus, and Viridans streptococci. And then for nasopharynx, again, Viridans streptococci, Staphylococcus aureus. And the reason I have bolded some of these are the ones that are clinically important, so you should definitely know know them for sure. And this is important because, so let's say somebody has an infection and you find Staphylococcus aureus in the nose, it doesn't often mean that it's an infection. So it could be part of flora, but if you detect it, you can kind of anticipate, because not everybody will have it as part of their flora. So, it's good to screen patients just to see if as part of their flora they had Staphylococcus aureus. So if you have infection in other parts of the body, you can kind of anticipate that Staphylococcus aureus found its way to other sites to find an infection. So perhaps the lower part of the respiratory system causing pneumonia, and we will talk about those scenarios throughout the course. Let's see. So the anaerobes in the nasopharynx are the same anaerobes as the mouth, and there are some gram-negative aerobes in the respiratory tract. So H. flu, Haemophilus influenzae, and Neisseria meningitidis is one. And then in the vagina, there are flora. For example, E. coli is a resident of vaginal tissue, and group B, Streptococci, and Listeria. Of course, when people mostly talk about flora, they typically talk about the GI tract, so gastrointestinal tract, commonly referred to as gut flora, and there are a lot. So you can see on the right-hand side that because of the harsh acidic environment of the stomach, not many bacteria can live in the stomach, and that's in general a defensive mechanism. And as you go down the GI tract, it's still acidic throughout, but the acidity decreases down through the small intestine and then the large intestine. So you can see as the acidity decreases, the number and variety of different flora increases. So especially in the large intestine, you will have a diverse body of flora, and this is a good thing, right? So the more you have these healthy organisms in your gut, they prevent through competition other problematic organisms from growing. So you're less likely to have an infection in the GI tract. Most commonly what we worry about is Clostridioides difficile infection. So because these organisms will compete with C. diff, it's healthy to have a healthy number of flora here. And this is also the reason why we are very careful when we choose antibiotics to treat other infections, because pretty much majority of antibiotics will affect the normal flora. So we want to avoid unnecessary antibiotics in order to preserve the flora. And when we cannot avoid antibiotics, we want to make sure that we use them for the shortest amount of time in order to minimize the collateral damage caused by antibiotics. But so you can pretty much see that these are mostly Enterococcus faecalis, which resides in the GI tract. Different species of Clostridium. A lot of these are anaerobes because this is an anaerobic environment for the most part. But some aerobes can survive here too. So Enterococcus is definitely aerobic and can have a big role here, as well as members of Enterobacterialis. So E. coli and Klebsiella certainly reside here. And the most important anaerobe is perhaps Bacteroides that resides in the lower GI tract, but pretty much unlikely to be found in other parts of the body. And in general, when people talk about antibiotics and ask if an antibiotic has anaerobic activity, they're primarily talking about Bacteroides rather than other types of anaerobes. Now, some important clinical concepts for interpreting cultures is, colonizer and contaminant. So anytime you have something growing in a culture, you need to ask yourself, is it coming from the source of the specimen, or did it get contaminated in the process when it was being collected? So a contaminant will not be from the site of the specimen. It could be due to improper technique, And then the colonizer is, if you detect something in your sample and it's not a contaminant, it means it came from the source of the specimen, but maybe perhaps it was not causing an infection. So a colonizer is essentially part of the flora, or if they didn't normally reside in the site as flora, but colonize it and it's not causing any problems. So that would be a colonizer. A good example is, some people may have Pseudomonas in the lungs, for example, and it may not, if they don't have any signs and symptoms of infection, it would be just a colonizer that normally would not be there in a healthy person. But, for example, someone with cystic fibrosis may have colonization of Pseudomonas in that region. Some other terminology is, when it comes to flora, is microbiota and microbiome. So microbiota essentially is all the organisms. So, and it could be bacterial, eukaryotes, and archaea, as well as viruses. So microbiota means any organism in there. Microbiome is talking about the genome of the gut. So it could be the genes that come from those organisms. That would be microbiome. And then there's the concept of prebiotic and probiotic. Probiotics are essentially live microorganisms that are ingested to aid health benefits. Prebiotics is essentially the food for these organisms. So prebiotics are in the form of fiber, and these are not absorbed by the body, so they will reside in the GI tract, and that would serve as food for these probiotics, so that they can thrive and basically lead to a healthy gut flora. When it comes to microbiota and probiotics, it's very tricky, so it's really hard to find for sure, which probiotic would be beneficial, if beneficial. And for the large part, it's because of individual differences in microbiota. So one person to the next, it's really different. The composition of microbiota is very different, and this is kind of part of why different individuals have different health outcomes. But it's it's really difficult to make sure that people have the same flora composition, and it may not even be necessary for everybody to have the same composition. But in general, we know that the more diverse the flora, the healthier the person would be. We interpreted the results of a specimen stain, described the process of microbiology testing for identification and susceptibility, and identify the normal human flora and their role in infectious diseases. Now let's explain the antimicrobial stewardship process for initiating, monitoring, and streamlining effective and safe antimicrobial therapy. Here's a systematic approach to infectious diseases. So when a patient presents with signs or symptoms of infections, or if they're suspected to have an infection, the first question you need to consider is, what are we treating? So this is a checklist of things to consider, and this, if you follow these steps in order, you are more likely to achieve the most optimal treatment for your patient. So the first question is, did you determine the infection site or type using subjective and objective patient information? So this is where you use the signs and symptoms. Then the next thing is to assess severity using SIRS or an infection-specific tool to determine where the patient should receive care. So in general, if a patient is more severe, they need a higher level of care, whereas if they're less severe, they may be able to be treated at home. So here, we go from low severity to high severity from left to right. So if the severity is very low, and the patient, for example, if the patient is ambulatory, so if this, if they are at the primary care physician or at the pharmacy or at the clinic, they weren't severe enough to be admitted to a hospital, so this will be treated as an outpatient. Now, slightly more severe infection, somebody, instead of going to the primary care physician or to their pharmacist, they may go to an urgent care to get higher level of care. And this still will be in an outpatient setting. So typically, a lot of patients will go to urgent care and they will go home with a prescription or perhaps receive treatment right there at the urgent care and then go home. And occasionally, they may have gone to the urgent care, and urgent care may, escalate it into an emergency department. So they may send them to the emergency department if that's appropriate. And you will see that emergency department at the hospital is kind of in between. It's the bridge between outpatient and inpatient, because a lot of people will go to an emergency department and then go home once they get treatment. So they may not be severe enough to be admitted to the hospital. So they may get a prescription and go home, or they may get treatment in the hospital and then go home. Or if they are severe enough, they will be assessed in the emergency department and then get admitted to the hospital, so typically in the general ward. And if it's more severe, they may need to be admitted to the ICU. So these will be in the inpatient setting. So that's why severity needs to be assessed in order to see where the patient needs to go. And then next is, did we obtain cultures from the infection site and remove, this culture, ideally we wanted to get to have it before we start antimicrobial agents in order to increase the yield of actually growing something. Because even a single dose of antibiotics before a culture is collected can result in nothing growing in the culture. Now, that's not always the case, but increases the risk. So ideally, if it's possible, we want to get the sample before we start antimicrobial agents. And then, of course, we want to make sure that we remove the infection source. And we'll talk about this source control throughout the course. And then the fourth question, did you list causative pathogens? And the reason this is important is because we are about to start empiric therapy. So before we actually find out what's causing the infection, we have to start treatment. We're not going to wait to find out. So you gotta have to make an educated guess. So it's important to list the causative pathogens that are most likely to least likely, so that we can choose empiric therapy to target those pathogens. And then the second thing to consider is, how can we ensure the patient receives the most effective therapy? So first we'll talk about effectiveness, and then we'll talk about safety. So for effectiveness, these are the things to consider. Obviously we want to start with non-pharmacologic therapy when possible. And then we want to use reliable resources, so guidelines. Sometimes there may not be a guideline for what the patient has, but typically we do have IDSA guidelines. So IDSA is Infectious Diseases Society of America, and sometimes other organizations may have guidelines, which we will cover throughout the course. And we want to make sure we choose antimicrobial regimen of choice based on guidelines or other reliable sources, such as randomized controlled trials. Next is, if more than one option is listed as first line, did we select regimen that meets the following criteria? So the first criterion is, did you use an antibiogram or another reliable resource to select a regimen with activity? And here's what an antibiogram is. So every hospital essentially has an antibiogram, and here's what it looks like. So over a period of time, somewhere between, at the minimum over a period of 1 year, so from January of December of the previous year, and sometimes you can even do it like for a 6-month period or even shorter, you essentially look at what grew throughout the year in the micro lab. So you, for each organism, as an example, I will use Acinetobacter. So over the period of 12 months, there were 55 isolates that were identified in the micro lab, and then you can see what was the percentage that was susceptible for each antibiotic that was tested. So if there's a dash, it just either wasn't active or wasn't tested. And then if there's a number, it's telling you what percentage was susceptible. So for example, here it will tell you that 22% of those 55 isolates were susceptible to ceftriaxone. So this number is percentage. It's not saying 22 of the 55. It's saying 22% of the 55. So these are pretty bad, as you can see, and that's because Acinetobacter is a problematic organism. And now let's compare that to E. coli. So you can see E. coli, there were 814 isolates for this time period, and 43% of them were susceptible to ampicillin. So that's pretty bad for ampicillin. But then if you look at imipenem, for example, you will see 100% susceptible. Or if you look at piperacillin tazobactam, you will see that 95% was susceptible. So that's how you use the antibiogram. So essentially, if you're empirically trying to cover E. coli, for example, you want to avoid ampicillin because there's only 43% chance that it will be active against E. coli, versus if you choose something like cefoxitin, that's a good number because it's a 95% susceptible. So a general rule of thumb is that for most patients, any number around 80% or higher is pretty good. Once the severity goes higher, for example, in the ICU, you don't want to take too much risk. So anything 90% or higher is pretty good in the ICU. But you have to use this cautiously. So you don't want to go specifically just pick the highest number. So it has to be within reason because there are other criteria to consider. It's not just these numbers. So for example, here between piperacillin tazobactam and meropenem, it sounds like meropenem is better choice because of 100%, but in practice, piperacillin tazobactam is a better option because it's a safer drug compared to meropenem because of the collateral damage caused by meropenem, which we will talk about throughout the course. Now, the next thing to consider is, did we use the active agent with the narrowest spectrum, which is kind of what I was alluding to when comparing, meropenem to piperacillin tazobactam. So the narrower the spectrum of activity, the less likely that an antibiotic will cause collateral damage. So that's an important consideration. And then the next thing is, did you use an antimicrobial agent that penetrates the infection tissue? So for example, if the infection is in the CSF, in the brain, you want to make sure that we choose an antibiotic that actually goes to the brain. It's not something that doesn't get to the site of infection. Or if it's in the lungs, in the respiratory tract, you want to make sure it's something that does go to the lung, as opposed to something that doesn't. And we'll again cover those throughout the course. And then the next question is, for severely ill, did you consider using IV antimicrobial agents? And this has to do with, should you use oral agents or injectable agents? And in general, when people are severely ill, you want to start IV first for several reasons. One reason is that it takes time for oral antibiotics to be absorbed. So you don't want to delay the action of antibiotics because you're waiting for absorption. So that's one reason. Second reason is, when people are severely ill, they are likely nauseated and they're vomiting. So you don't want to lose your antibiotics that you gave orally because of that. And then the third is that, depending on the illness itself, some people may not be able to absorb as much if you give orally. So for all of those reasons and many more, in general, you want to start IV when people are severely ill. Now, I do say severely ill. You don't want to start IV for everyone, but if they're severely ill, and then as they improve, we will have opportunities to switch from IV to PO. Now, the third thing to consider is safety now. So, of course, you always want to check for drug allergies and select antimicrobials that do not cause allergic reaction. So if somebody has a allergy to the drug that's listed, you want to use an alternative. For women of childbearing age, you want to avoid teratogenic, so anything category X you want to avoid. And then you want to evaluate if a patient is at risk for antimicrobial agent adverse effects, or if other contraindications exist. So check for any contraindication for each antibiotic. And then you want to check for drug-drug interactions. and depending on what other drugs the patient is on. For immunocompromised patients and some of the severe infections like meningitis and endocarditis, it's typically recommended to use bactericidal instead of bacteriostatic. And we'll talk about the differences between bactericidal and static throughout the course. And of course, another toxicity is cost toxicity. So if something is ridiculously expensive, we have to also consider cost. So you want, it's not that we want to go with the cheapest option. So we want to find a balance, between something that's effective, something that's safe, and also it's not unreasonably expensive if we have multiple options. And next we consider how can we ensure the patient receives the right dose. So once you select the effective and safe and affordable regimen, you want to make sure the dose is optimized. So typically you want to make sure if there's a need for a loading dose, and did you use the right, if it's dose per body weight, you want to make sure you use the right body weight depending on the antibiotic. You want to make sure that you adjust if it's needed for renal function and liver function. For severely ill patients, or if MIC data are available, we can use pharmacokinetic and pharmacodynamic principles to optimize the dose, and we'll talk about those principles throughout the course. For outpatient, we consider selecting a regimen dose no more than twice daily to improve adherence. So if you're giving an antibiotic that's dosed 4 times a day, that's very inconvenient, and it's easy for patients to miss 1 or 2 doses. In general, it's best to have something that's dosed once a day. That would be ideal. But typically 1 or 2 times daily is considered reasonable for adherence. And of course, you want to select the appropriate duration of therapy. In general, shorter is better. The more we study duration of therapy for various infectious diseases, we find that shorter is better. And the last thing is, how can we ensure this works and does not happen again? So we want to do some prevention. It's important to monitor for efficacy and safety, and this is typically we make sure that the signs and symptoms are improving, and also we want to look for adverse effects. And then if culture and susceptibility results have returned, did we start again at checklist item 7B? So that's where you can change to a more appropriate antibiotic. And the reason this is down on number 21 because it takes some time for these results to come back. So in the meantime, we did all the items on this checklist, and then when the results come back, we go back and start again at number 7B and go down. And then number 22 is, did you recommend immunization and other preventive measures? And this is if applicable. So for example, if somebody had influenza and you treated influenza, you want to make sure that you recommend that they get the flu shot the next time it's available, to make sure they don't get the flu again. And then last is, did you provide counseling to the patient about their infection and their antimicrobial agents? So you don't want to just only talk about the antibiotics, but you also want to talk to them about their infection. So you want to tell them what to monitor as far as signs and symptoms of infection improvement, right? How long does it take for their symptoms to go away, and what to do if their symptoms get worse, right? So sometimes if there is treatment failure, they may get worse. So you want to provide education to patient for that. This concludes this presentation.